is a website where we already have transcriptional maps, EQTLs available for everybody to mine. It's very compelling. When we started NEPTUNE, we expected to have the majority of our patients driven by a monogenetic disease, and that clearly is not the case. It's a small fraction, depending where and how you ascertain your patient population, between 6 and 12% as we recruit into community, so don't have a referral bias in our studies. It's 6.8% who have known monogenetic causes. A majority of those are collagen 4 variants. That excludes the APOL1 high-risk allele, which obviously is highly frequent in our patients of African descent with FSGS or nephrotic syndrome.
Thank you for that wonderful talk. So I have a general question for you. I really enjoyed learning about the TRPC6 story. So, I think the idea of moving into a blood or urine-based biomarker rather than a tissue-based, or translating this into something that's not as invasive is so important. You showed the example of urinary CTGF in TRPC6. My sense is that's actually a very generic biomarker. It's there in renal fibrosis and acute tubular injury. So I wondered, in your experience, how specific do you think we can get with any of these blood and protein-based biomarkers? It's not like we're going to use urinary CTGF to, attempt to treat people with the TRPC6 inhibitor, for example. So I always felt there can be a disconnect there, but you guys have done so much work in this space. So I was curious to hear your opinion and thoughts.
So it's a combination. We have baseline response predictors, which are the clinical profile, and they do reasonably well. ROC curves with an AUC of 0.74, 0.76 in that range. Adding CTGF push stats to the high 70s, adding urinary nephrin to 0.82 for you non-invasively predicting the intrarenal activation signature. Those activation signatures are not disease specific. That's exactly the point. This is where we always get the disconnect. TRPC6 activation, I do not believe, is something which is only seen in FSGS. What we cover with these non-invasive surrogates is an intrarenal cellular state, which can be targeted by the interventions, and can be, and we will have to show, and we expect the samples to be shipped to us, hopefully after Thanksgiving, to actually see how we performed. Did we indeed enrich in the trial population responders versus non-responders? Only a fraction of the patients in the TRPC6 inhibitor trial were recruited via NEPTUNE match. The majority of the North American patient was, but there was significant recruitment across Europe. So we have plenty of patients who came in without an activity assay. So we have patients with high and low activity in the study. And that obviously is a litmus test to see if the activity signatures enriches for treatment responses. In the endothelin receptor blocker setting with sparsentan, we have the data in hand, and indeed our sparsentan activation signature measured non-invasively, again, with pretty broad molecules where you would not expect them to be very specific for molecular mechanism, was indeed able to enrich.
Well, I think we're going to shift gears. I think we'll close grand rounds, but I just want to thank you. We had folks in this room from computational biology, pathology, adult nephrology, genetics. I mean, this is exactly what we were hoping to accomplish today. So thank you for everybody who joined us online, and for everyone who's here. We'll close grand rounds, and now we're going to shift into the next session. Thank you. Okay. Welcome all of our panelists up front. Fabulous. Yes. By all means. Yeah, let me ask Ingrid to help. Alan, do you have slides? No, no worries. Nope, some people do, some people don't. All good. Fabulous.
Overview of what we do.
Yeah, exactly. And I think you do.
Yes, do you mind if I, is this your computer?
Yeah.
Oh, do you mind if I?
No, 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 don't.
Appreciate it.
Yeah. I just got to tell you that septic, that was the most incredible grand rounds they've ever heard. I'm like, correct. Yeah. We'll have each person say something about what they do in the field, work in general, and some of the slides.
Maybe I'm the only one with slides, but.
Yeah, it's fine. I had thought about having some PEDSnet slides, but we've done other sessions on PEDSnet.
How do I share this, you know? Like, how do I?
Ingrid or Serena, can we get a little tech help up here? Just for this, do you mind for a second? Just in terms of the AV.
I need her to stop sharing.
So maybe while we're getting set up, I'll just say a few words of introduction. So in addition to being chair of the Department of Pediatrics, I'm the director of the Maternal and Child Health Research Institute. And what we have found to be incredibly powerful for MCHRI, which now has over 1,400 members, including members from every clinical and basic science department, as well as all the schools across this campus. We're just trying to marshal all the energy on this campus and get people to really think about maternal and child health. So this is the third time where we've had a gift, a really extraordinary gift from a visionary donor, where we have used MCHRI as the engine to create a whole community. So we had, the first one was in 22Q, the second one was in IBD. And I'll just use as an example, we now have 35 people on this campus who are funded through MCHRI to do IBD research, who were not doing work in IBD before this gift existed. And from every discipline you can imagine, the diagram's really quite extraordinary. Of all the disciplines that we've pulled in. There's some obvious ones, but there's also some really unexpected ones. And that's what we're trying to accomplish here with kidney disease. So in talking to the donor, the model we're using is that we do an RFA for grants to fund, these are $300,000 grants, to fund research in kidney disease with the hope of building an incredible community on this campus across many, many disciplines. And holy cow, was that grand rounds an extraordinary demonstration of what you can do when you bring all these disciplines together. So this panel here will speak to, I mean, obviously you've just heard from Dr. Kretzler, and I'm sure we all have some more questions for him, as I certainly do, in thinking about the next steps in ancillary studies. But we just wanted you to be aware of some of the things that already exist on this campus and the people, the clinicians, the scientists that are doing work in the field. So we're using this panel then discussion as a chance to introduce them to you more broadly. So Vivek is our pathologist. I'm going to welcome you to say a few words. One other thing I want to say is that what's going to happen moving forward as part of the Kidney Innovation Program is we're going to have a scientific advisory board. And so pleased that Dr. Kretzler has agreed to be on it. We also have Michelle Denberg, who's a pediatric nephrologist who's leading much of the work in kidney disease through PEDSnet, and Matt Sampson, who's a pediatric nephrologist and geneticist at Boston, who has an R01 that adds whole genome sequencing to all of NEPTUNE. So we're going to have, and then we're also talking about a couple other areas of expertise we might want to include in that scientific advisory board. So this is kicking it off, but there will be an annual scientific symposium where they will come out. The people who have been funded through this mechanism will present their research. Our scientific advisory board can advise us on where the field is going, and then we'll bring in keynote speakers. So really delighted to kick this off today, but please stay tuned. There's going to be a lot more really exciting activities on campus as relates to pediatric kidney disease. All right, Vivek, do you want to tell us a little bit about, I think this will work. Great.
Hi, everyone. Is the mic on? Can you guys hear me? No, right? Let me see. Okay, is that any better? Yeah, great. Hi, everyone. I'm Vivek Charu. I am a renal pathologist and a researcher here. And yeah, I don't know if you want us to do some quick introductions or if you want me to go ahead. Yeah, that sounds great. Yeah. So my hope today is just to share with you some information about some of the kidney biopsy tissue and biobank resources that we have here at Stanford. So the first thing I'll talk about is really our tissue-based resources based on our clinical practice. So we have a pretty large volume in-house and consultation renal pathology practice here, and our group, four of us, Neeraj Akamam, John Higgins, Megan Troxel, and myself, read all of those biopsies. Some of them come from in-house, but many of them come from, all over the Bay Area. So for all of these biopsies, as you may or may not know, there is a core of tissue that's put in formalin, and then we have remnant FFPE blocks of those tissue. And as Dr. Kretzler, demonstrated in his wonderful talk, there's a lot of new technology and techniques that we can use FFPE tissue for. We also have some remnant frozen tissue that was originally used for immunofluorescence microscopy. Some of those tissue, depending on the year, will have been lightly fixed in Michelle's, and some of it will be directly fresh frozen. So we save all the tissue, going back 20 plus years. So we have a large volume of these types of cases. Our group has expertise in cohort creation, tissue microarray, TMA construction. I'll show you some examples of that. We have been developing some pipelines for spatial transcriptomics in collaboration with Brooke Howard in pathology and Giuseppe Barsano in the QSU. We also run our clinical testing, on a whole genome backbone for monogenic kidney disease or inherited kidney disease. We have also established a research pipeline for whole genome sequencing with Stuart Scott in pathology at Jan Gwen in the QSU. So just to give a quick example in what is possible with some of our tissue-based resources. So in a 6-year period from 2015 to 2020, we made the diagnosis of IgA nephropathy in 791 patients. Of those 791 patients, there's 415 patients with linked longitudinal records. So this gives you a sense about the scale, for a common glomerular disease of what might be possible in terms of linking our existing tissue with outcomes and clinical phenotypes in these patients. And I'll just give you some sense about what might be possible using FFPE tissue or things that we have been doing via an example. So, we have been interested in looking at gene expression profiles in minimal change disease and in TIP-bearing FSGS. We can form a tissue microarray in which we sample, needle core biopsies of multiple patients and put them on a single slide, and we can perform spatial transcriptomics in this group. This is an area where cohort creation and case selection becomes very, very critical. And this is something that anyone in our group would be very happy to help people with. So if there are questions, about what tissue-based resources we have, or there are ideas about how we can use some of these tissue-based resources, please reach out. This is just an example of some of what we're doing with the Cosmex. We are able to get good cell segmentation, excellent cell type calling. We use, of course, the KPMP as a reference for a lot of this work. And so this just gives you a sense that in a single slide, we can profile, 113,000 cells and 136 million transcripts. And we can, really start to get into in-depth characterization of cell-specific gene expression and cell-cell interactions. This is just one technology that we happen to have a lot of, interest and expertise in, but, there are so many others, and we'd be very happy to work with anyone in this way. The second area is really a longitudinal kidney biopsy biobank that we have been organizing. And this work is really funded through a collaboration between pathology, Tom Monty, adult nephrology with Tara Ching, and pediatric nephrology with Scott Sutherland. And it's Amanda Tapia is our main CRC who organizes this effort. For all patients undergoing kidney biopsy at Stanford, they're consented to provide urine and blood. So far we've enrolled, accrual is continuing, we've enrolled around 130 patients. Nearly all of these patients provide blood, and we separate the blood into plasma, PBMC, serum, and whole blood. We have multiple aliquots on each patient. Obtaining urine is actually a little bit more challenging for whatever reason. It could be because of the timing of when the consent is happening, but we only have urine on 25 patients or so. You can see the breakdown of this cohort, in the table. And, obviously we hope to expand this effort, but, we do have a mix of native and transplant biopsies. I think from this table, it seems like there's probably a good opportunity to use some of these samples in the setting of transplantation, probably, studying rejection phenotypes and others. But of course, if someone wanted to do a very deep dive in a particular disease, we do have some available resources in this way. I think maybe the best way of potentially using some of these resources is either to do some kind of discovery and potentially look at NEPTUNE and CureGN as validation, or vice versa, things like that. I'll also put a plug for the Human Immune Monitoring Center, which is run by Holden Maker, and his contact information is here. So, they do routine assays on both blood and urine. Here's just a sampling of what they're capable of doing. I spoke with him, before this talk, and I think there's a lot of opportunities for, using some of the blood and urine that we have already and performing some of these assays. So with that, I'll stop. Please contact me if there's any interest in using material from our biobank or using material from our tissue. Our historical clinical samples. I think any of our renal pathology group would be very delighted to collaborate with anyone. Thank you.
Going down the line, Matthias, could you say a few words about how people have used all these resources for career development awards? And MCHRI funds master's tuition for fellows that want to get a master's in clinical research and epidemiology. And then we have a lot of physician scientist pathway programs. Has anyone ever used these resources to write an F-32? I'm assuming there's K awards that are funded, but could you just say a little bit about the career development opportunities through this consortia?
Obviously, that's critical, as we feel that the clinician scientist is actually the rate limiting step. It's not data, it's not patient, it's the clinician scientist who's connecting the dots. If any clinician scientist is in the room, yes, that's you. Yes, do not worry. I know we have a lot of background noise about, what is the fate of, research. You are heavily sought after wherever you are in your career, because the opportunities are unprecedented and the resources behind that are significant. So please stay the course, but connect with research efforts like the one you are seeing presented here today. We have very strategic efforts in the space. We have a young and mid-level investigator community who meets monthly between NEPTUNE and CureGN. No, senior investigators are not allowed. This is where you get together and you share all the backstories about what worked and what didn't work for you. At our NEPTUNE investigator meeting mid of October, we had an entire morning dedicated to actually two topics. One topic was young investigators, research paths, opportunities, funding, networks, connections, how to accelerate your career. The other session was on digital pathology and AI. There's a very rich community, and the research networks I presented this morning are a great source for data and information without the need to spend a few $1 million of generating these data sets. And as I stressed, those are open science communities and KPMP, truly openly downloadable to you. You don't have to talk to any of us. But you want to for two reasons. You know the data, and we can tell you, challenges and opportunities. And B, we will connect you. We will connect you to people who do cutting edge science. And the reason why I'm thrilled to be yesterday and here today here in Stanford is that, yes, senior investigators listening in, we will connect you. And you might see young people knocking on your door and saying, look, I have those cool opportunities in the kidney space, and I saw your papers and your stuff on GitHub. Can you let me know how I can modify and adjust that? But that's strong recommendation. Please connect with those frameworks wherever they are. All of us know that we will roll out the red carpet for you to ensure that you are asking the right question together with the right people.
Yeah, thank you. And just for all the folks who are online, just want you to know that I'm monitoring the Q&A. The other thing we should have mentioned is that this gift includes substantial support for biostatistics. So Manisha Desai, who runs the quantitative sciences unit, couldn't be here this morning, but was at the dinner last night but she is going to make sure that there are faculty within the quantitative science unit that become really deeply knowledgeable about all of these resources, all of these opportunities, where they're going to have point person who will then be the biostatistician dedicated to this work on campus, who our hope is will go to the NEPTUNE and the NEPCURE and the CureGN and really meetings become deeply embedded in with a deep content expertise around what's available through PEDSnet and available through all these consortia. So please know, as you think about the projects you want to do and you think about the grants you want to write, we all know that sometimes the analytic costs can be daunting, but we have resources available specifically for that as well. All right, Alan, you want to talk to us about PEDSnet?
Sure. Okay. Can we hear? Yes, we can. So my first disclosure is I am not a nephrologist. So in fact, I was thinking as you were talking about the last time I actually took care of a patient with nephrotic syndrome, and it's been a while, which is, since I mostly work in the PICU, it's really a tribute to our nephrology group and how well they manage their kids, since they don't make it to the PICU very often. I'm a clinical researcher. I do some work in UTI, and so I'm interested in the relationship between UTIs and structural abnormalities and chronic kidney disease. And I've been the PI for PEDSnet for about 2 years. PEDSnet, for those of you that are not familiar with PEDSnet, it is now a 13 Children's Hospital collaboration. It's over 10 years old. Started at CHOP. It's an EHR, a comprehensive EHR collaboration where we all funnel our EHR data on all of our patients that we see both on the inpatient and the outpatient side into a common data model, and that is used. It has been used historically primarily for clinical research, comparative effectiveness type questions, but increasingly we are moving towards trying to be a true learning health system. One example, maybe one of our intensivists here is doing a implementation science project to try to get people to transfuse less in the pediatric intensive care unit. That's an AHRQ PEDSnet funded project. There has been a strong track record for kidney-related work. Mary already mentioned Michelle Denberg, one of Mary's longtime mentees as well, and she's done a considerable amount of work looking at bone health. Now, actually, is creating a user interface for chronic kidney disease that will be housed on the PEDSnet website, which is pretty cool. So all patients with chronic kidney disease in PEDSnet, you can sort of anyone can access that data and get sort of basic demographic and some clinical data on patients with chronic kidney disease in PEDSnet. We've been part of PRESERVE, which is PEDSnet is one of, I think, 4 PECORNet hubs in the PRESERVE study, which is sort of trying to look at interventions to prevent progression of chronic kidney disease in children. So I can say more, but that's a brief overview of what we do. I see Shaina in the audience here. She has some funding through MCHRI within PEDSnet to look at sort of kidney outcomes in critically ill patients who have had some AKI during their PICU stays. So we're very excited about that work, which is ongoing.
Okay, thank you. Sophia.
Thank you. Good morning, everybody. I'm Sophia, one of the pediatric nephrologists here. I wanted to add also for the PEDSnet data, so Michelle Denberg also has helped to define clinical phenotypes through PEDSnet, and so that does make it easier to do research in the glomerular space for kids with glomerular disease. And so I wanted to put that out there as well, that they do have pretty well-defined phenotypes out there, and it's a really great chance to use real-world data and do research on in that space. My special interest is in lupus nephritis, and so my kind of background is that I have worked with industry, kind of using data sets from industry to look for predictive markers of response to B-cell depleting therapies in lupus nephritis. I am leading the clinical side of this program, and so in addition to all of the great research that is going to be done, hopefully through this program, we're also trying to build out in the clinical space collaborating with rheumatology to create joint clinics, trying to improve transitional care for patients with glomerular disease, and also kind of trying to strengthen our existing connections and create new connections with other or just national efforts. And so I'm here to help provide the clinical correlates to all those who want to do research to help improve outcomes in children with glomerular disease.
Let's see. Yes, good morning. Nice to see you. I'm Rich Lafayette. I'm an adult nephrologist, and the ushering in of this age of pediatric further immunologic glomerular disease research is just music to my ears. I've been trying to move the dime a little bit since about 20 years ago when I sort of washed up a little bit administratively, being Senior Associate Chair of Medicine and Clinical Chief of Nephrology for a long time. Sort of want to get back to the patients and seeing that we were just marching in place and not really making advances, and said we had to do better, again, particularly in glomerular disease, where, as mentioned, we were basically in the adults just giving steroids to everybody, getting them sick and not seeing responses. Was working with my pediatric colleagues who are doing the same, but adding on Cytoxan CellCept to try to get better responses, and seeing these very sick 16 to 20-year-olds present to my clinic, not any better. So, really been trying to make these efforts for a long time. At Stanford alone, I got my own development gifts, went to some of your senior investigator labs at the time, 15, 20 years ago, and said, what do we do here? I have this very specific disease. And they said, just do omics, just do omics. And I said, okay, let's do omics. So I started a biobank, and we still have about 150, 160 old blood and urine samples for genetics, omics, BBMCs, urine, that still could be used. They're in deep freeze with well-categorized patients. But once I met Matthias and said, okay, we can do this more nationally and globally, that made a lot more sense to develop much more rich data sets to really let professional omic people describe what's going translationally. And I moved into trials more and more, and now we organize multiple global trials across glomerular diseases. We partner with pediatrics intermittently and hopefully much more routinely now to really bring in patients and, as said, get all the information we can from them, all the biological data as well as clinical data, and make sense of it. So I think there's just a huge opportunity now with all the information that's in the Stanford medical record, in Stanford patients, in these global registries, and collaborative databases, that any scientific question can be asked and the preliminary data can be risen. And with funding now, it's just a no-brainer that we can move forward. And of course, I always say this is a model because we have all these things in glomerular disease of antibody-mediated kidney disease, complement-mediated kidney disease, genetic-mediated kidney disease, and general progression models that lead to fibrosis. And these things are common to whatever organ, whatever patient problem you want to address. Ultimately, there's cross-reactivity. So hopefully we can all work together and see your hypotheses and see whether we can fund them and make patients' lives better. So thank you.
So much. Well, when we were in our conversations with this donor, Rich, we really highlighted your work. Vivek, we highlighted your commitment in this area. So all of you have really played a meaningful role in helping secure this gift. And then just so you can see up on the screen are the details, the seed grants. They're $300,000 each. We were sort of thinking R21-ish, rounded up. And we're going to be funding 3 a year for the next 5 years, and really excited about the community this is going to build. Do we have any questions from anyone for anyone on the panel? Yes, please. Come to the microphone. Or yeah, thank you. And I want to thank the MCHRI staff who coordinated this session, as always.
Thank you. As a data scientist and AI person, I had a few questions with respect to the data sets. It sounds like between NEPTUNE and the Stanford data sets, we have a total of about 2,000 patients, would you say? between these total about 2,000 patients, and for them we have coordinated data in NEPTUNE for some of the omics studies that I saw. And in here, you have in the PEDSnet, we have much larger data sets for broader studies as such. So that's the total. And in terms of techniques and problems that have already been addressed on these data sets, from a data science point of view, is there a repository or information about that? Like what problems have actually been addressed? Because some of the techniques I saw today, like use of self-organizing maps, which is one of the classical methods in data science, but maybe that makes sense because the data sets are small, but in terms of what studies have already been done from a data science perspective, that would be good.
Yeah. So the good news is that the different data frameworks from the bottom up have used all the same models. So NEPTUNE and CureGN, they are coordinated by the same people with different funding streams. NEPTUNE patients roll over from NEPTUNE if they have completed 3 to 5 months of follow-up into CureGN for hopefully indefinite long-term follow-up. CureGN has 2,800 patients. NEPTUNE has touched 1,200 patients, 946 where we have the phenotypic information available. The subsets for the deeper molecular genetic informations are subsets of these patients. We have a transmart instance, which is a data mart where you can dynamically explore the data sets, the overlap, which data elements are available, which characteristics, as a nice cohort building tool to see where to go. I would invite you, if you have ideas and concepts, to bring them forward. This is such a rich data space, and you're absolutely correct. We started to work with self-organizing map 11 years ago. I never would have expected that to be an old technology, but it certainly is. And we had fun discussions yesterday throughout the afternoon, with all the different resources you have available with model systems to extract knowledge in unsupervised and supervised approaches. So I would not consider any of the problems solved to an extent that it would not warrant engagement of data analytical person in the setting.
Yeah, and I would just add that CureGN is equally excited about the work we're doing here. I've had conversations with Bill Smoyer, who's the PI of CureGN, as well as I had some conversations with their scientific advisory board when we were creating the proposal for this gift. So in the RFA for these funding opportunities are links to both the NEPTUNE and the CureGN ancillary study sites. So those are just two among many other opportunities. Matthias, you had a slide, I think, that showed there's just a growing number of consortia with data available. So thank you.
Nice.
Yeah, Vivek.
Yeah, and I just wanted to add regarding the Stanford resources. So, the existing biobank and our tissue, clinical tissue repository, we have not generated data from those. My hope or my dream is that some investigators will be interested in generating new data from what we have, and that can contribute or add in a way to the CureGN and NEPTUNE data that's already out there. But I think it's very important to also try to generate new data in addition to what's already out there.
Yeah, I also wanted to add that for CureGN and NEPTUNE, because the proposals for the ancillary studies, they go through reviewers, and there are opportunities where, because there is a lot of overlap between NEPTUNE and CureGN, and so they are looking at patients with with the same diseases, except for CureGN has an additional one, which is IgA nephropathy. But when you do submit your proposal, then reviewers will look to see whether there are opportunities to leverage data from both data sets.
Those are a continuous effort. I would not consider them competitive in any way, shape, or form. They approach patients at different stages. In NEPTUNE, we approach patients before they undergo a biopsy, so you get a true source population with deep tissue level information, but the sample size is a third of what now is available in CureGN, and CureGN has more population level definition of disease burden, long-term outcome, and associations. They are coordinated by the same people. Laura Mariani, when she came to Michigan, had a K award in NEPTUNE, and I was the initial data coordinating center Contact PI, which transitioned to LAURA 3 years ago. We have our offices wall to wall, so we are a continuum, and the mechanisms are reflecting that. There's also a pilot funding mechanism available from the NEPTUNE Foundation as well. Every other year, we fund 2 grants with a similar amount as you just have shown. So there are also independent funding mechanisms available, and being a CureGN or NEPTUNE auxiliary study investigator is good for you. The funding rate is over 30% from the various NIH funding mechanisms.
So I just wanted to follow on Vivek's idea of generating ideas, new ideas locally from local biorepositories. So in this age of national studies with these huge sample collection, what do you think the value is of local biorepositories, A, and B, whether some of this gift funding can be leveraged to fund these biorepositories? Because unless you do it right, you're not going to collect enough samples in a meaningful way.
Yeah, I think this is really critical. All the protocols are publicly available, how samples are procured, processed, and stored. And this has been utilized across Europe, even in sub-Saharan Africa with the Kidney Disease Reach Network from H3 Africa, we are using the same protocol. All these studies are independently governed, give plenty of opportunities for investigators to drive their science forward and establish leadership roles. But if we continue to do that, to ensure that the way sample and data are generated is comparable, then those studies can be mutually supportive. And that's why I think we have also protocols publicly available, so it's known what happens where and when. Those are usually low intensity protocols because they have to work across 32 sites or in the jungle of Africa. Okay? So what the opportunities for centers of excellence like Stanford is that, you know, as you have deep immune phenotyping capabilities, which are clearly not scalable across 32 sites, that you leverage those expertise, that you have samples processing activities where you need immediate access to samples to, you know, bring them through, you know, genome scale data generation activity. An example for in that space was during the pandemic, we wanted to quickly monitor how the kidney responds to a COVID infection. And we established a protocol where we are doing single cell sequencing out of urine samples. But for that, you really have to have fresh samples who in a matter of 45 minutes are in the machine. So we were not able to keep these cells alive sufficiently enough to ship them between centers. So in similar aspects, you know, phosphoproteomics of, you know, peripheral blood cell populations, those are opportunities clearly where centers of excellence like yourself will be driving the science forward and then establish the same reference samples the larger networks have. And yesterday, obviously, you have beautiful people who then can even take those into EHR-derived knowledge environments like PEDSnet, if you have, again, enough anchor data sets to walk across so that you can take from your 12 patients where you have deep immune phenotyping over 15 patient visits in your setting, project them into data spaces of cohort studies and then into the EHR national registries.
Yeah, I want to thank you for the question. It's a really important question. The IBD gift, which was on a larger scale, did include $1 million a year for biobanking and built a very robust biobank. This is a new foray for us in kidney disease. It was because we didn't have the ability to build that infrastructure so quickly that early in the development of this proposal, we reached out to NEPTUNE and CureGN precisely because we knew that would be important. But we have very ambitious plans for what we're going to do in kidney disease here, and we'd love to build out more of our biobanking infrastructure. So thank you for the question. Oh, back here, good.
Thanks to the panelists. My name is Noah. I have a background in sickle cell nephropathy, but currently working primarily on stem cell regenerative medicine, hematology focus. I've heard a lot of clinical work or vision toward this award or grant. I wonder if there is a lot of space for basic science and translational, and if you guys can comment on the investment of this award toward that type of research.
Absolutely. I mean, emphatic yes. We're very much interested in that kind of work. I will say there's a separate part of the gift carved out, a different funding stream to support the work that Alicia Bertani is doing with Paul Grimm. And so the dual immune solid organ transplant, there's whole separate, very substantial amount of money that's funding that work. So you might want to have some conversations with Alicia about what they're doing on this in terms of stem cell transplant and regenerative medicine. But we very, very much embrace fundamental discovery and the sort of things that could be done at the fundamental basis and then validated through things like NEPTUNE and Cure. I'm sure, Matthias, you probably have some examples about how critical that's been.
Yeah. I think, and one of the exciting pieces is that, you have the firepower of Stanford available here. But what's different from 15 years ago when we started, you now can take your basic science insight and rapidly accelerate into clinic. Yeah? And I hope even made it clear that you can do the inverse as well. You can use the clinical trials, which Lafayette is designing with the key trialists in the country and globally as a discovery engine for you as well. And that, I think, what has changed. So the basic clinical walls, from my vantage point, they are gone. It's just us and foundational support as we have now available here in Stanford to make sure that that valley of death is, a very fertile area of exploration.
Great, thank you for the question. Okay. Let's see, I was tracking online. I don't think we have any questions online. Nope. All right. Any final questions? All right, well, I just want to thank everyone who joined us. There's a lot of new faces in this room. There's a lot of new names online. This is exactly what we were hoping to accomplish. Really, really grateful for the role that all of the panelists played in helping secure this wonderful gift. And we know we're gonna execute on it and deliver everything that we promised. And all of you will be an important part of that. So thank you to everybody.